Up on the second floor of Rigby Hall is a lab where junior biology majors are busy learning basic procedures on how to research different genes. This story will be following the endeavors of Keaton Sowers and Heidi Grable as they work on such things as polymerase chain reactions and gel electrophoresis. Dictystelium and E. coli cells are being used as model organisms to help us find out different things about genes, which can in turn help us with our own DNA. This is a project where I myself have also worked on with these DNA samples and have now passed them on to the year below me.
My name is Heidi Grable, and I'm a biochem major. I believe, I've talked to Dr. Lindsey many times, and every time I do it, I just feel stupid. That what I'm doing is I've got dictystelium cells, I took the DNA from the dicty cells, and now I'm looking at two different genes. I think it's only one at this point. So we amplified that gene through PCR, and I think we're going to make some plasmid or something with it. I am running a gel to test and see if my PCR worked at all. Loading the wells is pretty tense because you don't want to get it too far down, and you don't want to put a hole in your well. So you want to get it all very nicely into your well, and you have to be very focused, very steady. Okay, so after we ran our gel, we went and looked at our gel under the UV light, and we got bands, and it looks great, and we thought that we did amazing. Come to find out, the bands we got was just the control, and that was supposed to work. So I went to talk to DL, and he's like, yeah, so the primers don't work. So this week, we're trying a whole bunch of new primers. We are redoing the PCR, which we've already done. This would be our third time doing PCR this quarter. It's only week 4. And now we're running a gel at the moment, and hopefully it will work out. So, it is now almost 6 hours from when I started this lab. And we just got our results back. And we were hoping that despite making the mistake, it would be okay and we'd still get the results that we were looking for. However, the mistake we made was we put ladder into our samples. And the ladder is the only thing that showed up in any of our samples. So none of them worked, once again. This class is useful because I want to do research for genetic diseases and try to cure them. And to do that, I need to be able to do things such as PCR, gel electrophoresis, Western Blots, all of these kinds of things. So when I go to grad school, I'll be one step up on the others because I actually know how to do these things already. I am cutting out a certain gene, and the primers can recognize this sequence of base pairs, so the adenine, guanine, thymine, and cytosine, and they attach to it and cut it out. So by the end of your PCR, you have a lot of that one gene. DL started this lab doing it this way a couple years ago when he decided that this would help students when they graduated. And the way it works is when one year finishes, the next year picks off where the year before left off. So the lab just keeps going based on what the previous year found out. So therefore, if people messed up with the primers or the DNA 2 years ago, then that still carries over to the year that we're in now.